Okay, so protein structures, we have four different levels of protein structures. We have the primary, secondary, tertiary, and quaternary structures. For each structures, mayroong particular characteristics. We have, for primary, it's all about sequence. So, sequences of amino acid actually makes the primary structure. For secondary, we have the local structures. So local structures here means that combination of primary structures to make the secondary structures. Okay, so if we combine primary structures, we can create the secondary. For tertiary naman, it's the overall three-dimensional shape. Nakakaroon ng parang 3D shape sa tertiary when we combine two secondary protein structure, for example. Then we have quaternary. We have subunit organization. This quaternary structure will help later on keeps a protein's shape, and of course it will help sa protein para magawa niya yung function niya. Bakit kaya kailangan ng structure for proteins? Kung i-sequence natin lahat ng amino acids of a protein, sobra siya kadako. So, dako kayo siya or taas kayo siya. That's why we need to a structure ng protein in which we tends to fold their structure para masigo po siya sa isa ka area or kanang para mahimo siya kanang compact. Kaya kung i-sequence nato lahat, taas kayo ang structure nila. Aside from that, also protein is needed to have a perfect structure because the shape ng protein also affects the function. So, sa biology kasi, the shape of a molecule is very important for them to be able to do their function. Without this shape, that's the time na medyo naipagka-problem sa
So, makulat lang ko sa inyong question.
So that's the time na medyo na magka-problem ang proteins if wala sa siyahang perfect shape. So the shape is necessary for the protein to do its function. So how can we create that particular structure of a protein? Ginagawa natin sila ng folds. By folding the protein, you can actually create the shape of the protein. So we have here the primary structure of a protein is actually determined by covalent bonds, which means we can do primary structure by doing covalent bonds. Then we have secondary, tertiary, and quaternary structures are actually determined by weak forces. Weak forces here are the following. We have the hydrogen bonds, ionic interactions, van der Waals interactions, and hydrophobic and hydrophilic interactions. Meaning, sa composition pa rin ng proteins, may effect din siya sa kanilang structure. So for example, sa proteins natin, di ba mayroong mga polar tsaka non-polar, right? So those polar proteins, they tend to be more excited to meet water. So they love water. That is why mostly those mga polar amino acid nasa gawas. While yung mga hydrophobic na mga amino acids which are non-polar, sila itong nasa sulod na part sa protein structure. So that's one way of knowing, or that's one way of affecting the structure of protein based on weak forces. So ito yung itsura ng apat na structures of proteins. So we have the primary, these are chains of amino acids. Then we have the secondary, the tertiary, and the quaternary. So as you can see, protein is actually made by condensing the amino acids together to make such shape.
Tapos,
So all of these are made up of amino acids. Remember, di ba, yung ating amino acids our proteins actually or peptides starts with the N terminus and usually ends in C terminus. Kaya mayroong mga NC dito na nakalagay. It means dito nag-start yung protein and ends at the C terminus. So, yan, another structural differences of the four protein structures. Again, primary protein structure is a sequence of a chain of amino acid. So, ito yung primary structure. So, each of this circle represents an amino acid structure. Okay? And of course, they are bonded by peptide bonds. Now, yung dalawang primary structure, mag-bind siya together through hydrogen bonding. And this hydrogen bonding will tend to make those primary structures fold into two ways. So, pwede siya mag-fold via a plated sheet like this one or alpha helix like this one. Pwede siya may mong duha. Possible na siya na yung duha. Then, the tertiary protein structure is done when the side chains naman ang mag-interact. Ano pala yung mag-interact dun sa secondary? Yung kanilang peptide backbones. Yun yung nag-react sa secondary. While sa tertiary, ang nag-i-interact naman will be the side chains. Then sa quaternary protein structure, the quaternary naman is the combination of two or more tertiary protein structures. So this will be the last structure of a protein. So the primary structure, again, sequence of amino acids, and it starts with the N terminus and ends with the C terminus. When you say N, ito yung parang part ng amino side or amine group. Well, yung C naman is a carboxyl group. The numbers of amino acids vary. So, for example, we have the following amino acids. We have the following primary structures. Insulin, for example, is a protein that have 51 amino acids present in that chain. Lysozyme with 129, hemoglobin with 574, and gamma globulin with 1,250 amino acids. So, mag-vary na siya. The primary structure also determines the folding of the polypeptide to give a functional protein. So the primary structure is also dependent on the type of amino acid present in the chain, like polar amino acids and non-polar amino acids. So we have polar amino acids. We have three types: acidic, basic, and neutral. So polar lang to siya. Then we have the non-polar. And the polar amino acids are hydrophilic again. So they are mostly placed on the outside of the protein. While yung mga non-polar naman na mga amino acids tend to be placed on the inside of the protein para makaiwas sila sa presence of water. So this is an example of a primary structure with the chains. So again, it starts in the N terminus and ends with the C terminus. So this is a 25 amino acid primary structure protein. So napakadaming amino acids present. And they can be combined together to form different kinds of proteins. Since maraming function yung proteins natin, so marami siyang possibilities of combinations. Now, importance of primary sequence. There are many protein diseases involved in the primary amino acid chain. So an example of that is sickle cell anemia. Sickle cell anemia is a disease that causes our red blood cells to shrink. And this is due to the changes in the protein structure, primarily in their primary structure. So, if you can see the example, we have the following amino acids. We have DLS, PADK. Pagdating dun kang sickle cell, may changes between kang D tsaka kang V. So na, instead of D, naging V na siya. So parang ganyan yung idea na once na mayroong problem sa kanilang protein or sa kanilang chain, there would also be a consequence for them na maapektuhan po ng atuang bodily structures. Okay, so yan yung example of importance of the primary sequence. So kung na problem with that, with the combination of the amino acid, it will create a disease. In this example, we have the sickle cell anemia.
Or wala.
D, yung D pala sa hemoglobin refers to aspartic acid. So instead of aspartic acid, naging ano siya, valine. So, kailan ba nagkakaroon ng changes sa amino acid na sequence? The amino acid kasi can be combined based on the data na nandun sa DNA sequence. It's important for us to have a correct DNA na sequence para correctin yung protein na ma-translate ng mga ribosomes. Kung mayroong mali doon sa DNA sequence, that's the time na mayroon ding mali sa protein na sequence. So, kung may mali doon sa protein sequence, yan, may problem na doon sa ating body. So, those are some examples. So, again, since we have a lot of amino acids and we can combine them in whatever position na meron, as long as correct na position siya, the number of possible sequences is infinite. So an average protein has 300 amino acids. At each position, there could be one of 20 different amino acids. So if you're going to know exactly ano yung possible combination, so it's 10 raised to the power of 390 possible combinations. Sa secondary structure naman, so ito yung example. So the secondary structure is the folding of the NCC backbone of the polypeptide chain using weak hydrogen bonds. So if you can see the example dito, we have yung connection ng blue. Ito yung mga hydrogen bonds.
Anyway, so siguro guys, ay mga kanil may lain yung mga written task in the first quarter. Ay, ayun na lang napiling ko. May lain yung written task for first quarter.
Yeah. So these are the hydrogen bonds of, let's say, part sa amino acid na structure. And those hydrogen bonds makes sure na mag-fold sila na ganito.
So, isa pa lang.
So, for example, isa siya ka-chain, right? Then we try to fuse this side or this side. So, kung i-fuse na to siya, di ba may mo siya ginana?
Okay, nakalimutan ako sa mga kauban.
So, that particular folding now will be used as the secondary structure. So, once na napod siya kauban na isa pa, or combination of primary structures, so, mag-create na siya ginana na itsura. So, parang ganyan sa secondary structure. Again, hydrogen bonds. So, may bond na nangyari dito, tsaka dito. So, again, we have two types, di ba, of folding for the secondary structure. Yung alpha helix and yung beta plated sheet. So, yung alpha helix, yung sample natin kanina, yung nakaganon.
Okay.
While yung sa beta plated sheet naman is parang paganon. So, wala siya papel nga gi-fold, no? So, parang nana si beta plated sheet. While yung katong Alpha helix naman is gilukot siya na papel tapos gituyok na wala inani. The length of the helix or the peak is determined by certain amino acids that will not participate in these structures. Example, we have si proline. So, ito na yung example natin kanina na may isang primary structure and another primary structure. So, within the primary structure, ang nag-combine sa kanila kay ang peptide bonds, right? But with the other peptide, ang nag-combine siya kay ang hydrogen bond. So, para madikit nato ang duha ka primary structure, need natin ng hydrogen bond. Si alpha helix, this is the alpha helix, is a right-handed spiral. When we say right-handed spiral, yung kanyang turn is from right. So, para siyang paana. So, that's the right-handed spiral. Residues per turn is 3.6. The residues here means yung amino acid na nag-combine doon sa ating chain. Once kasi na you add one amino acid, then another amino acid, then tanggalin mo yung water sa kanila, we can call those as residues. So, each amino acid residues, ang kanyang size is 3.6. Then their repeat distance is 5.4. On average, there are 10 residues per helix. Average lang yun siya, so possible na lesser or more than 10. Amino acid side chain points out from the helix. When we say point out, it means nasa labas sila. Kumbaga na, example man ito ang helix. Kumbaga na nasa gawas ang mga side chains. So that's the alpha helix structure. Then we have the beta-plated sheets. So sa beta-plated sheets naman, again, it's a secondary structure wherein we have to use two primary structures. Dapat they lie adjacent to one another. When we say adjacent, dapat magkatabi. So our groups alternate, first above, then below the plane. The CO and NH groups, or the carboxylic and amino group of each peptide bond, are perpendicular to the axis of the sheet. So we have two types of beta-plated sheets. We have the anti-parallel and parallel. So si anti-parallel, ito yung example natin. While si parallel, ito. Kung anti-parallel, different directions. While kung parallel, same directions. So, depende na siya sa type of amino acid present. Kung mag-alpha helix ba siya or beta-plated sheets. So, for example, if ang protein is primarily beta-plated, na siya'y high content of alanine and glycine as their amino acids. So, depende na gina siya. Which means that a structure of a protein will also depend to the sequences of the amino acids. Kay laylay ba silang side chain, no? So, kung laylay silang side chain, maglaylahi po na sila o connect. This is how the secondary structure differs. So, possible siya nga inani, alpha helix, or possible siya nga beta-plated sheets. For the tertiary structure naman, it's the folding of the polypeptide into domains whose chemical properties are determined by the amino acids in the chain. Again, si amino acid has a factor for the tertiary structure. Example, we have the milk protein. So, ganito yung itsura niya. So, sa tertiary structure, this is more on a factor ng mga weak na relationships or interactions. So for example, sa tertiary structure, the folding is sometimes held together by strong covalent bonds, right? So mayroong strong covalent bonds. Also, mayroong disulfide bonds na nangyayari dito sa tertiary structure. The bending of the chain takes place at certain amino acids like proline. So kung makita na nato nga naa dito si proline, possible nga dito siya mag-bend yung chain. So the way we arrange themselves para maging tertiary structure, of course, is based doon sa interaction ng mga amino acids. So for example, si hydrophobic ug si hydrophilic mag-start nag-fold. Si hydrophobic tends to go inside of that tertiary structure, while si hydrophilic naman is more on the outside. Since they are water-loving and water-fearing amino acids. So this is an example na, let's say this is our alpha helix na secondary structure. So alpha helix daw to. Then meron pa ring hydrogen bonds, disulfide bridge, and ionic bond between the side chains. Okay, side chains na to. Remember, side chains are free sa primary and secondary structure. So, mag-combine sila later on sa tertiary na structure. So, these are the factors that will make the tertiary structure. So, maraming factors when it comes to tertiary structure. So, possible na yung mga side chains ng amino acids are hydrophobic. So, sila ang mag-ato sa tunga. So, di sila ma-expose sa gawas. Possible po na magkaroon ng hydrogen bond between the two side chains. So, possible. Pwede po magkaroon ng disulfide bonds like this. Or ionic bonds. So, an example, you have here the primary structure. Si primary structure, possible siya mahimong alpha helix or beta plated sheet. So, si alpha helix, for example, of one primary structure, then si beta plated sheet with another, pwede siya mag-react with one another by reacting their side chains, and thus making the tertiary structure. So, this is an example of a tertiary structure for protein kinase. So, if you can observe, we have the alpha helix here, and we have the beta plated sheet here. And they are combined, of course, they are connected via different bonds. Then lastly, for the structures, we have the quaternary. Some proteins are made of several polypeptide subunits, like hemoglobin, for example. Meron siyang 4. And this illustration on the right is an example for protein kinases. So how can we determine it? So si protein kinase, may dalawa siyang tertiary structure. Ito yung una, tertiary structure, at ito yung pangalawang tertiary structure. Combine them together to create a quaternary structure. So sa quaternary structures naman, they are made up of the tertiary structure, which we call subunits. Those subunits fit together to form the functional protein. So therefore, the sequence of the amino acids in the primary structure will again influence the protein structure at the secondary, tertiary, or even the quaternary structure. That is why very important na tama ang pag-code or pag-translate from DNA to protein. Okay, once na mali sa chain of amino acids, magkakos na siyang disease sa to. Yan, so this is also the a structural difference between the structures. So yan nga, we have the primary structure here, which are made up of different amino acids. Then, ang Mga-cause for us to make a primary structure, kailangan natin ng peptide bonds. Sa secondary naman, kailangan natin ng hydrogen bonds. So, anong nagre-react dito sa kanila? Ang nagre-react dito sa secondary are the amino group or the carboxyl group. Then, si tertiary structure, need natin ng connection ng kanilang side chains. So, sa side chains, possible yung ionic bonds, hydrogen bonds, disulfide bonds, hydrophilic tsaka hydrophobic interactions. Those are the important na mga interactions to create the tertiary structures. So, we have examples of proteins. We have collagen and hemoglobin as this example. So we have collagen, which is a tertiary structured protein, while hemoglobin is a quaternary structured protein. So again, the quaternary structured proteins are a combination of the tertiary structured proteins. Another example is the hemoglobin molecule. Again, hemoglobin is a protein present in the red blood cells, and it is a tetramer composed of 4 polypeptide subunits. So when we say tetramer, it's made up of 4 polypeptide. So we have 2 alpha subunits and 2 beta subunits there. And present also in each of those subunits are the Hem group, which is the iron portion of the hemoglobin. And that Hem group can bind 4 molecules of oxygen. So in each hemoglobin molecule, we can bind 4 molecules of oxygen. So this is the hemoglobin structure, and it's the chemical formula of hemoglobin. Another examples are immunoglobulins. Immunoglobulins are antibodies produced by the plasma cells or the white blood cells. They are highly specific and has a memory, which means that if the body of a human is infected with a virus, for example, that was already introduced to the body, it can remember kung ano yung action na ginagawa niya doon sa virus na yun. Also, the antibodies can recognize self from non-self, meaning they can recognize ano yung cells na normal sa body at ano yung mga foreign cells. So those are the characteristics of immunoglobulins. So in their structure, they contain 4 peptide chains that are connected by disulfide bonds and arranged in a Y-shaped quaternary structure. So we have here an example of a chemical structure of immunoglobulins. So again, they are shaped into a Y-shaped na structure, and we have the disulfide bonds here. The SS means disulfide bonds. So those disulfide bonds is the reason why the immunoglobulins look like a Y. So this is another example of the structure of immunoglobulin. So the red portions are the disulfide bonds. Then each of these structures are the different chemical components of immunoglobulins. So result, again, protein structure depends upon the amino acid sequence. If there's a problem with the amino acid sequence, there's a problem with the protein structure. And if there's a problem with the protein structure, there's a problem with their functions. Because for a protein to be able to do its function, they must retain their shape. Once that their shape is broken or their shape is changed, so they cannot anymore do their function. So for an amino acid sequence to be correct, the sequences of the bases in our genetic material must also be correct. Because when we produce protein, we base that sequence from the DNA sequences that we have. So if there's a problem with the DNA sequence, it would also trigger a problem with the amino acid sequence. Next, we have classes of proteins. For the classes of proteins, we have 5. The fibrous proteins, globular proteins, simple protein, conjugated protein, and dietary proteins. So for simple protein, it's a protein composed of only amino acid residues. Example, we have ribonuclease with 124 amino acids, and as well as albumin. It means wala silang other kasama na molecule. It's purely amino acid residues. That is why they are called simple proteins. Conjugated proteins, on the other hand, are proteins that incorporates one or more non-amino acid units in its structures. So here we add another molecule aside from protein to create the conjugated protein. Examples, we have glycoproteins, lipoproteins, and metalloproteins. So glycoproteins, for example, is a molecule, protein molecule combined with glycogen. That is why it's called glycoproteins. Lipoprotein is a molecule that is a combination of protein and lipids. That's why it's called lipoprotein. So those are the example of conjugated proteins. You have also fibrous protein. They are tough, insoluble, and they are composed of fibers and sheets, and they are involved in the structure of a cell and also contractile movement of the cell. Example, collagen, keratin, elastins, myosins, fibrin, cilia, flagella, and the mitotic spindle. So basically, si fibrous protein, sila yung responsible again for the cell structure and for movement. Collagen, for example, is present sa cell membrane, as well as a cell wall of mga plants, while flagella are used by the sperm cell for them to move, or even for bacteria so they can also move, while mitotic spindle is used to do or to help in the mitosis and meiosis na process. Another example of the fibrous protein is keratin. So keratin has an effect on why some people have curly hairs. So example, keratin is rich in the cysteine residues that form disulfide bonds cross-linking the adjacent polypeptide strands. So since marami siyang disulfide bonds, it creates kumbaga bonds with the other polypeptide strands. So a permanent curly hair, for example, involves reducing and oxidizing the disulfide bonds to change the degree of curl or wave. So ito yung permanent. So this permanent na curly hair again involves reducing and oxidizing. A set is yung parang iset mo lang yung hair mo, hindi siya permanent na curly hair. It's wetting and drying, of course, and it rearranges the hydrogen bonds naman between the helices and fibers. So in humid days, a dynamic rearrangement of hydrogen bond is occurring when hair becomes frizzy. Globular proteins. These proteins are usually found sa cell membrane. They are water-soluble and have chains folded into compact shapes. So most globular proteins are moving around the cell. Example of this globular protein is the albumin, also casein in milk. The globular proteins are also the type of proteins with binding sites, meaning they are able to bind other molecules. So we have Enzymes, for example, as an example for globular protein, as well as hemoglobin, immunoglobulins, and membrane receptor sites. Then we have dietary proteins. Dietary proteins, these are proteins that are needed by our body. So we have actually 4 types of dietary proteins. So we have essential amino acids, which makes up the essential proteins that we need. Also the non-essential amino acids, the complete proteins, and also the incomplete proteins. So essential amino acids are those amino acids that cannot be produced naturally by our body. That is why they are called essential. Kasi hindi natin siya napoproduce. While yung non-essential amino acids naman, those are the amino acids that can be produced by our body. Complete proteins are derived from animals, and it provides all of the essential and non-essential amino acids. While the incomplete proteins naman are derived from vegetable sources. So, incomplete ang tawag sa kanila because they lack a sufficient amount of one or more essential amino acids. While comparing it with the complete protein, we have the exact amount of essential amino acids as well as the non-essential amino acids. Now, for protein functions, of course, proteins serve different functions inside our bodies. Again, the function of proteins is based also to the structure of proteins. So once that their structure is damaged, of course, their function also will not be the same. Denaturation or inhibition, which may change protein structure, will also change its function. Coenzymes and cofactors in general may enhance the protein structure. So if mayroong denaturation na nangyayari, coenzymes and other cofactors might be able to help in enhancing the protein structure. So the way we classify proteins, we classify them based on their structure, or we classify them based on their functions. So we have the classification of proteins by function. We have the catalytic proteins. Catalytic proteins are proteins that can catalyze mga reactions. So example of those proteins are enzymes. We have also storage proteins. Storage proteins are of course used for storage of mga molecules. Example, we have ovalbumin for eggs, casein in milk, or zein in maize or corn. We have also transport proteins. An example of a transport protein is hemoglobin, which can transfer oxygen from the lungs to the other parts of our body, and as well as carbon dioxide. We have also communication proteins, or proteins that can do communication. So one example of that protein is hormone. Hormones, example of hormones is one example of hormones is insulin, and as well as neurotransmitters. Neurotransmitters are found sa atong mga nerve cells or neurons. So they are responsible in communicating with the other nerve cells para ma-process sa atong brain stimulus, para maka-react tayo doon sa stimulus. Contractile proteins are proteins that can contract, of course, and this is usually used. These proteins are usually found sa mga cells wherein it allows movement. So, for example, we have actin, myosin, and dynein, which are usually found sa mga microtubules. We also have protective. Protective proteins are those proteins that would help us fight mga foreign cells or even mga foreign materials. Examples of protective proteins are immunoglobulin, fibrinogen, and blood clotting factors. There are also proteins that are used as self-defense. So for the self-defense, we have toxins. Toxins are usually found sa mga animals na merong venom, for example, snake. So snake venom is actually a toxin. So it acts like a self-defense ng mga snakes. While we have also structural proteins that can also help to maintain the structure of a cell. Example of this structural proteins are keratin and collagen. We have diseases that can be caused by an abnormal protein folding, meaning may deformation doon sa protein structure. So if mayroong deformation, that might cause diseases. So one example is sickle cell anemia. So sickle cell anemia is a disease wherein mayroong deformation of our red blood cells. And this disease was caused by the deformation of hemoglobin, which is present sa atong red blood cells. So if that red blood cell, that hemoglobin, a protein found in our red blood cells are deformed, meaning it can also cause the deformation of our red blood cells. And with that, it can cause the sickle cell anemia. So those are some examples. Then we have also Alzheimer's. Alzheimer's disease is due to the deformation of the protein called beta amyloid peptide. It can cause plaques in neural tissues. Then we have also Creutzfeldt-Jakob. With affected protein niya is the prion. Same lang also with Alzheimer's. So yun yung mechanism niya. We have also hereditary emphysema and cystic fibrosis. So ito yung mga affected proteins and ito yung mechanism why they are affected. So for denaturation of proteins, it occurs when the organized structures of a globular protein becomes completely disorganized. So we have the following denaturing factors. First is temperature. So as the temperature increases, it also increases the rate of molecular movement within the solution, and the bonds within the proteins begin to vibrate more violently. So when it undergoes violent vibration, the bonds in that globular protein, for example, becomes broken. That is why ma-disorganized po ng ilang shape. So what will happen? They lose their characteristic three-dimensional na shape, and of course, they will become completely disorganized. So anong temperature ang needed to denature proteins? So we have 56 to 60 degrees Celsius or higher. That is the temperature that is needed for us to be able to denature proteins. Coagulation occurs as the protein molecules then unfold and become entangled. So they have aggregated to become a solid. So once that they are disorganized, ma-disorganized sila, there will come a time that they will also do coagulation. So once na mag-coagulate na sila, they will become more of a solid na shape niya. Dili na siya ma-change. O maglisod nag-bind dito. Let's say yung mga enzymes, for example. Maglisod na siya o bind. Muna dili na nila mabuhat ilahang work. So here is an example of the denaturing factor, which is the temperature. So we have alpha helical proteins in a solution, and when we introduce heat to that solution, it will cause intense vibration that will again destroy the structure of the alpha helical proteins. And then they will become denatured. So we have this one. And there would come a time that they will do the coagulation. So once that they will be now coagulated, ito na yung itsura nila, and they will not be anymore be changed into alpha helical proteins. So this shape now is not the same with this shape, meaning they cannot do anymore their purpose, which is dapat magawa nila if this is their shape, yung alpha helical. In denaturation of proteins, the structure na hindi kaya i-denature or hindi kayang i-break down ng mga denaturing factors is the primary structure. Meaning they can break down the secondary structure, the tertiary structure, and the quaternary structure, but not the primary structure. pH is another denaturing factor. When the pH of a protein solution is above the Pi, all the protein molecules will have a net negative surface charge. Below the Pi, they will have a net positive charge. So in either case, these like charged molecules repel one another, and this repulsion helps keep these very large molecules in solution. At the Pi, the protein molecules no longer have a net surface charge. As a result, they no longer strongly repel one another and are at their least soluble. So that's how pH can affect the protein structures. So here is an example. So we have a molecule with a net charge of positive 2 and a net charge of negative 2. So once na maging neutral na ilang Pi, so wala na silang net charge, this will be now the time na medyo least soluble na yung mga molecules. Organic solvents naman are also a denaturing factor. So we have polar organic solvents that can denature proteins by destroying their hydrogen bonds. So by destroying their hydrogen bonds, they will of course change the structure of the protein. And in addition to forming hydrogen bonds, the solvent, which is the water of course. Detergents naman have both a hydrophobic region and a polar or hydrophilic region. Detergents are another denaturing factors, and they interact with proteins. How would they interact? They disrupt the hydrophobic interactions within the protein, causing the protein chain to unfold. Remember, hydrophobic interaction happens inside because they are hydrophobic. It means they are afraid of water. So detergents, for example, will go to that interaction sa loob, and they will destroy or disrupt those interactions. So what will happen? The protein will unfold and exposing their, of course, other interactions doon sa kanilang structure, causing the change of their shape. Heavy metals like mercury or lead are again another denaturing factors. They may form bonds with negatively charged side chain groups. So when they bond with the side chain groups, this interferes with the salt bridges formed between the amino acid or group, resulting in loss of conformation. So dito, yung exposed na side chains, which are exposed, can be bonded with a heavy metal, and when magband na dito si heavy metal, ma-change na ilahang shape. That will cause the denaturation of proteins, for example. Mechanical stress also, like steering, whipping, or shaking, can disrupt the weak interactions that is present in a protein that maintain their protein conformation. So may weak interactions doon, 'di ba may weak and strong interactions tayo sa proteins? For weak interactions, mechanical stress is enough for them to become disrupted. So ang simple stirring or shaking lang can actually weaken the bond or the interaction. Of course, that will cause for the disruption of the protein structure.